José Antonio Ainsa, who is associate professor at the University of Zaragoza. He has a long history of work in microbacteria, and having worked also at the Institut Pasteur in France and in Norwich, United Kingdom, and his presentation is on antimicrobials, discovery, development, and mechanisms of resistance.
Okay. Okay, thank you very much. I want to thank the organization for inviting me to present an overview of just one of the works that we have done in our group. I'm the leader, along with the next speaker, who is Santiago Ramón García. We are the principal investigators of the antimicrobial groups at the University of Zaragoza. So, as I say, I'm going to start by saying very obvious things. You all know that all human beings are different, and we can think of many examples of it, no? So, from my point of view, I'm a microbiologist. What I want to tell you is that for bacteria, this is true as well. So, not all strains of Mycobacterium tuberculosis, for example, which is our favorite bacteria, the bacteria in which we are working, not all strains are the same. So genetic studies that have been done during the last decade have shown that there are very different genetic lineages within the species of Mycobacterium tuberculosis. And this has very important implications from the point of view of tuberculosis, because one of these genetic lineages is the so-called the Beijing family of strains. And these are characterized by being more transmissible, and they are prone to acquire multi-drug resistance very frequently. So for this, we need tools to detect specifically the strains, the Mycobacterium tuberculosis strains of the Beijing family. So a few years ago, the work of one of my PhD students, we discovered a single nucleotide polymorphism which was specific for most Beijing strains. And this is the sequence that we could find in the non-Beijing strains, which is the site for our restriction enzyme. And in the Beijing strains, we found that there was a C inserted in here, which destroyed the cleavage site for the restriction endonuclease. So this was very, very important. We were very excited about this because this allowed to think of three potential three options for detecting this polymorphism. One is the most obvious. If you get a PCR product, you cut with this enzyme. If the enzyme cuts the PCR product, that means the strain will be a non-Beijing lineage. But if the PCR product is complete, is not cut by the enzyme, that will mean that this is a strain of the Beijing lineage. Another option that we developed more recently is to develop specific oligonucleotides that will only amplify a PCR product in case that the strain we are studying belongs to the Beijing phenotype. Okay, so we thought that this could be helpful for clinicians to help them to detect specifically the Beijing strains, but however, these two methods are very easy, could be very simple for a basic research laboratory like the one we are working, but however, for a clinician, should not be very helpful because that means extra work to do a PCR, to run an electrophoresis, and so on. So because of this, we have thought about other kind of assays like the so-called line probe assays, which are very common for detecting, for example, drug resistance in mycobacteria. These line probe assays are the ones in which you have some strips of a material in which there are some proofs that has been unmobilized, and you can hybridize, and depending on the pattern, specific pattern of hybridization that you get, you can detect whether one strain is multi-drug resistant or not. So our vision is to produce a new kind of product in which using the polymorphism that we have detected in the Beijing strains, we could have some more proofs in here that could help very easily at the same time that the clinician detects for drug resistant, they could detect whether this specific strain belongs to the Beijing genotype or not. So this is something we have patented, and what we would like, this is our dream, this is our vision, is to look for help for any potential partner, a biotech company, or any other that could help us to develop this product. And if so, we will be very happy to collaborate with you. So as I say, my name is José Ainsa. This is my email address if you want to contact me. And the final slide, as I say, we are the antimicrobial groups of the University of Zaragoza, along with Santiago Ramón García, who is the other principal investigator and who is going to introduce an overview of the whole group. And we have to thank the present members of the group and also our supporters and our collaborators. So thank you very much. Any question or next speaker?
Dr. Ainsa.
Okay.
I suggest we just proceed with the next
Yeah.
speaker.